Hello everyone and welcome to today's session, and today we'll be discussing the practice questions related to the chapter evolution. After this we'll also discuss the mock test. Okay, so this is the disclaimer. So what are the objectives of today's session? So today we'll be discussing the basic concepts which are related to the chapter evolution by walking through the NCERT practice questions. Okay, so these questions, they are based on certain concepts. We'll discuss these concepts by walking through these practice questions. Okay, so we'll be discussing NCERT practice questions today related to chapter evolution. Let's begin with the first question. So first question is, according to Charles Darwin theory of evolution, the main mechanism of evolution is. Okay, so you have four options A, B, C, D. You have to choose the correct answer from these options. The first option is mutation, second is natural selection, third is genetic drift, and fourth is gene flow. What will be the correct answer? So let's have a look at the answer. See, option number B, natural selection, is the correct answer. Let's have a look at the explanation. So Darwin proposed that natural selection is the main driving force of evolution. This means that nature selects which traits are favorable for survival and reproduction. Okay. Now it works through survival of the fittest. Okay. So natural selection works through the idea of survival of the fittest. So this doesn't necessarily mean the strongest, but rather those best suited to the environment, they will survive. Okay. So the organism which is able to adapt to the change in the environment, that organism will survive. That organism is fit. It will produce more number of offerings because it will be selected by the nature. Okay. Organisms with favorable traits are more likely to survive and reproduce successfully. Okay. Clear? So advantageous traits, they are being selected and they become more common in a population over time. Clear? Now, as a result of this, we observe a gradual change in the population over time. The gradual change is what we refer to as the evolution. Let's discuss next question. So next question is, which of these represents convergent evolution? Okay, so let's have a look at the options. So option number A is wings of butterfly and bat. Option number B, thorns of bougainvillea and spines of opuntia. Option number C, flippers of penguin and fins of the fish. And option number D, all of the above. What will be the correct answer? So option number D is the correct answer, all of the above. Let's have a look at the explanation. So convergent evolution, it occurs when different organisms, means unrelated organism, they develop similar features. Okay, because they are under similar environmental pressures. The environment plays a significant role in shaping the adaptations of the organisms. Now in convergent evolution, the organisms, they are unrelated to each other. They are not closely related. They are unrelated species, but they are developing similar features. Okay, why they are developing similar features? Because they are exposed to or they are under similar environmental pressures. Now let's understand this with the help of examples. Okay, so this is an example. Convergent evolution, you can say, leads to the formation of analogous structures. Analogous organs or structure means they have functional similarity, but their structure and origin is different. Okay, so butterfly, okay, wings, then bat's wings, bird wings. Okay, their structure and origin is different, but their function is same, which is flying. Clear? Now let's move on to the next question. The evolution of Darwin finches is an example of. So what are the options? Option number A, adaptive predation, B is genetic drift, C is convergent evolution, and D is parallel evolution. So what will be the correct answer? Okay, option number A is the correct answer here, which is adaptive radiation. So adaptive radiation, okay, it is the evolution of different species from a common ancestor. Clear? Means there is a common ancestor, and from the common ancestor, what is happening? Different species are evolving. Clear? All of these different forms, okay, they are adapting to their own environment. Okay. A very classical example of adaptive radiation is Darwin finches. So all of the Darwin finches, they evolved from a single species. Okay, and this occurred on Galapagos Island. So all of these different Darwin finches, they evolved on Galapagos Island. Galapagos Island was geographically isolated, and this provided the perfect setting for adaptive radiation to occur, leading to evolution of the new species. Okay, so Darwin finches, okay, they develop different beak shape and sizes to exploit different food resources. Okay, so this is a very clear example how physical traits can evolve to meet specific environmental challenges. Now, this led to the formation of multiple species adapted to different niche. Okay, each species which is found in its own ecological role and is demonstrate the power of adaptive radiation in creating biodiversity. Okay, so each species is adapted to its own environment. Each species has its own ecological role. Okay, each species has adapted to different niche. Okay, so this leads to biodiversity because it is leading to evolution of a new species. Here you can see in this image, these are the different beak size and shape in Darwin finches. Now next question, in the Miller-Urey experiment to demonstrate chemical evolution, which combination correctly represent the atmospheric gases and conditions used? Okay, so these are the four options here, A, B, C, D. You have to choose the correct answer from these options. Clear? So these options, what do they represent? They represent the primitive conditions. Okay, conditions present in the primitive earth. Okay, so what will be the answer? Okay, the correct answer is option number B. Okay, methane, ammonia, hydrogen, water vapor in the form of H2 and electric discharge. And the temperature was very high, around 800 degrees Celsius. So Miller-Urey experiment used reducing atmosphere components. So Miller, he mimicked the conditions present in the primitive earth in the lab. Okay, so the key gases which were present in the primitive earth, they were methane, ammonia, hydrogen and water vapor. Clear? Now electric discharge was used to provide energy. So electrodes were used to generate an electric discharge. So this electric discharge provided energy for these gases to combine in various ratios. Temperature was very high, around 800 degrees Celsius. There was reducing atmosphere. There was no free oxygen. And these conditions, okay, there was, you can say compounds like amino acids, they were created. So we can say that by this experiment, these organic compounds, they can be created from the inorganic compounds. Okay, so this experiment produced organic compounds including amino acids like glycine. So this is a setup that was used by Miller. So in this flask, you can see in this bigger flask, he introduced all these gases. There was no oxygen. Oxygen was removed with the help of the vacuum. Then using this electrode, electrical discharge was created. This is a condenser, water-based condenser. Okay, and he observed the formation of amino acids. The next question, which scientist first proposed that embryonic layers of different animals show similar development patterns, supporting the evolutionary relationship? Okay, here they're talking about the embryology, development of embryo. So what are the options? Ernest Haeckel, Carl Ernst von Baer, August Weismann, and Hans Spemann. What will be the correct answer? So let's have a look at the answer. Option number A, Ernest Haeckel, is the correct answer. See, Ernest Haeckel proposed the biogenic law, also known as the recapitulation theory. So he stated that ontogeny recapitulates phylogeny. What does this mean? This means that an organism's embryonic development repeats its evolutionary history. Okay, so during the development of the embryo, what is happening? Ontogeny repeats the evolutionary history. Okay, so he made detailed comparative embryological studies of various animals. Now his work, though partially incorrect, was significant in showing how embryological development can provide evidence for the evolution. Okay, so Ernest Haeckel theory was actually rejected by Carl Ernst von Baer. So Carl Ernst von Baer said that during the development of the embryo, okay, this embryo will develop uniquely. It will not show you can say, parts of the adult form of any other species. Next question, the phenomena where a small population splits from a larger one and establish a new colony leading to reduced genetic diversity is called. So what are the options? Founder effect, natural selection, gene flow, or genetic hitchhiking? What is the correct answer? So let's have a look at the answer. Option number A, founder effect, is the correct answer. Let's have a look at the explanation. Okay, a small group, okay, it breaks from a larger population, which is a source population, and it forms a new isolated population. So imagine there is a one large population, which is the source population, and a group of individuals, they migrated to another area. Now this new population carries only a limited sample of the original population genes. Okay, clear? So because of this, now what will happen? The allele frequencies will change. Here, the new population has reduced genetic diversity, and this can lead to unique evolutionary paths. Here you can see in this image, this is the parent population, and a group, it breaks away from this larger population. So this is the migrating population, and then this, you can see, this is a population after a couple of generations, and in the end you have founder population after a few more generations. So in the founder population, the allele frequency is different from the original parent population. Clear? So this is known as the founder effect. Okay, the small group of population, that is known as the founder population. It will multiply, it will divide. Their alleles will increase in number. Clear? Now let's move on. Now genetic diversity is reduced in the new population due to this limiting sampling. So small group of individuals migrated from this larger population, so obviously they will have reduced genetic diversity. Okay, so we can see allele frequency in the new population may differ significantly from the original population. Clear? Here in the end you can see in the founder population, the allele frequency is different from the next question, when natural selection favors extreme phenotypes at both ends of the distribution curve, which selection against intermediate forms it results in? So you have the option. First option is direction selection. So second is disruptive selection. Okay, option number C is stabilizing selection. Option number D is random selection. What will be the correct answer? Option number B, disruptive selection, is the correct answer. So disruptive selection occurs when extreme phenotype, extreme traits are favored over the intermediate traits. So this results in bimodal distribution in the population. So you will see two peaks here. Example are African finches, where either very small or very large beaks survived better than those with medium sized beaks or average sized beak. Here you can see in this image, image C, this is showing you disruptive selection. Here you can see there are two peaks, extreme phenotypes are being selected instead of the intermediate phenotype. Okay. So this type of selection can lead to speciation over time. So the original population was split into two distinct groups with different phenotypes. Now next, which of the following evolutionary forces actively changes allele frequencies in a population? What are the options? Random mating, natural selection, infinite population size, or no mutation? What would be the correct answer? Okay, option number B, natural selection is the correct answer. See, natural selection is an active evolutionary force that changes allele frequencies. Okay. Random mating, infinite population size, and no mutation are actually conditions required to maintain the Hardy-Weinberg equilibrium. Okay, so in this case, the allele frequency will remain constant over the generations. Okay, under natural selection, favorable alleles are selected by the nature. Okay, alleles which give survival advantage, they are selected. Okay, so the frequency of the favorable alleles will increase, while the frequency of unfavorable alleles will decrease. Clear? What are the other forces that can change allele frequencies? That is mutation, which is random changes in the DNA sequences. And due to mutation, variations occur. New traits can also, you can say, develop due to these mutations. So due to mutations, alleles can form, which are different form of the gene. So these alleles can provide different phenotype, different traits, which can be beneficial to the organism. Okay. Gene flow, which is transfer of gene between populations through movement and breeding of individuals from one population to another. The next one is genetic drift, which is random change in the allele frequencies. This is basically noticed in small population and this occurs by chance. Another one is non-random mating. So when organism chooses to mate, when organism choose mates based on specific traits rather than randomly, this affects the genotype frequencies or allele frequencies. So when a particular organism is choosing a specific mate, then only those alleles will be carried on to the next generations. Okay, moving on to the next question. What type of energy source was used in Miller-Urey experiment to stimulate light rings? Options are UV radiation, nuclear radiation, electric discharges, or heat only. Let's have a look at the answer. See, option number C, electric discharges is the correct answer. So, Miller, he used electrodes to generate this electric discharge to provide energy. Okay. Yeah, so electric sparks were used to stimulate the lightning in the primitive Earth atmosphere. So these electrical discharges provided the energy which was necessary to break the chemical bonds and form new organic compounds. So here you can see in this image, this is the setup used by Miller's. So here you can see he used electrodes to create the electric discharge which simulated the lightning in the primitive Earth. Okay, so you can see all these gases were added. So by this energy, chemical bonds were broken and new organic compounds were formed such as amino acids. Okay, this is clear. So now let's discuss the mock test. Okay, so first question is the anticodon loop in transfer RNA is responsible for. Okay, so we have 4 options. You have to select the correct answer from these options. The first option is binding to amino acids. Second is recognizing the complementary codon on mRNA. Third is initiating translation. And fourth is terminating protein synthesis. What will be the correct answer? Okay, option number B will be the correct answer here. So see, transfer RNA acts as a adapter molecule. Okay, so it is a link between the codons on the mRNA and the amino acid. So it brings the corresponding amino acid. Clear? So it recognizes the codon on the mRNA with the help of the anticodon loop. Clear? Option B will be the correct answer. So next one, which of the following is not a goal of the Human Genome Project? Okay, the Human Genome Project, what was the focus of Human Genome Project? To sequence the human genome. Okay, now what are the options? Address ethical, legal and social issues. Then option number B, determine the sequence of all the 3 billion base pairs. Option number C, develop methods for human cloning. And option number D, improve tools for data analysis. What would be the correct answer? The correct answer is option number C, develop methods for human cloning. Okay, so rest of the options given here are true except option number C. This was not the goal of Human Genome Project. Clear? So human cloning, this is something different. Okay, this is done in biotechnology. Okay, so this was not the aim, to develop methods for human cloning, cloning of human genes, or human cloning as it is. Next one, what are SNPs in the context of human genome? What are the options? Are they repeated DNA sequences with no coding function? Option number B, single nucleotide polymorphism indicated base differences. Option number C, X-ray sequence tag used for gene identification. And option number D, microsatellites used in physical mapping. What would be the correct answer? Okay, here the correct answer will be option number 2, single nucleotide polymorphism indicated base differences. Okay, so SNP means singular nucleotide polymorphism. Means there is a difference in one base. Okay, there is a difference at only one position. Because SNPs are quite common among the individuals, and this is the cause of variation as well. Okay. Next one, what percentage of human genome codes for proteins? Okay, so most of the human genome, it doesn't code for proteins. It is known as junk DNA. But what proportion of human DNA codes for proteins? So options, first option is less than 2%, second is approximately 50%. Option number C, around 25%. Option number D, over 99%. What is the correct answer? Option number A will be the correct answer here. Less than 20% is the correct answer. So actually less than 20% of the human genome codes for proteins. Okay. So this is expressed into proteins, less than 2%. The rest is junk DNA, which consists of a repetitive DNA. Okay. Next one, what was the approximate total cost estimated for the Human Genome Project at the beginning? So here the options are 3 billion US dollar, 9 billion US dollar, 20 billion US dollar, or 1.4 billion US dollar. What is the correct answer? The answer is option number B, 9 billion US dollar is the correct answer. Okay, this was quite straightforward question. Let's move on to the next question. So question is, which chromosome in humans has the fewest gene according to Human Genome Project? So option are chromosome 1, chromosome X, chromosome Y, or chromosome 21. What is the correct answer? So here the correct answer will be chromosome Y. Okay, this is one of the sex chromosome present in males. So it has fewest genes. Clear? Next question. Which factor is responsible for terminating translation by binding to the stop codon? Okay, translation is what? Translation is through translation proteins are produced from the mRNA. Okay, mRNA is translated into proteins. Okay, so what is the name of the factor? Initiation transfer RNA, elongation transfer RNA, release factor, or ribozyme? What is the correct answer? The option number C will be the correct answer here. Release factors, they bind to the stop codons. So basically stop codons that do not code for any amino acid. Okay, so during the termination of translation, these release factors, they bind to the stop codons and this initiate the termination of the translation. Next, during translation initiation, the ribosome recognizes which codon to start polypeptide synthesis. Okay, so they are talking about the start codon here. What is the sequence of the start codon? So options are UAA, AUG, UGA, and UAG. What is the correct answer? Okay, option number B is correct answer. AUG, this is the start codon. AUG codes for methionine. Next one, what is the process of linking an amino acid to its cognate transfer RNA? What is the name of this process? Okay, peptidation, amino acylation, elongation, or initiation. What will be the correct answer? Okay, what is the name of this process? So option number B is the correct answer, amino acylation. Okay, this is also known as charging of the transfer RNA. So when amino acid, it binds to the transfer RNA, it is known as amino acyl transfer RNA. Process is known as amino acylation. Next one, what is the role of untranslated regions in the mRNA? Okay, options are they code for amino acid in the polypeptide. They are required for efficient translation. Option number C, they bind to the release factor during termination. And option number D, they form peptide bonds between amino acids. So what is the correct answer? The option number B will be the correct answer here. These untranslated regions, they are required for efficient translations. Okay, so these regions, UTRs, they don't code for any proteins. They are not translated into proteins, but they are very important for efficient translation. Next, what is the primary basis of DNA fingerprinting? Okay, so options are single nucleotide polymorphism, variable number of tandem repeats, which is VNTR. Third option is mitochondrial DNA variations. On option number D, ribosomal RNA sequences. What is the correct answer? Okay, option number B will be the correct answer here. Variable number of tandem repeats, which is VNTR. Okay, so certain sequences, they are repeated, and these sequences, they are adjacent to each other. Clear? Now the copy number of these repeats are different in different organisms. Okay, this is the basis of DNA fingerprinting. These are basically VNTs are basically repetitive DNA sequences. The next one, which scientists contributed to deciphering the genetic code by developing a cell-free protein synthesis system? Okay, so options are George Gamow, option number B Hargobind Khurana, option number C Marshall Nirenberg, option number D Severo Ochoa. What is the correct answer? Okay, Marshall Nirenberg is the correct answer. Okay, so he created cell-free protein synthesis system. Okay, and with the help of the system, okay, he was able to decipher the genetic code. Okay. Next one, which component of ribosome acts as a catalyst for peptide bond formation in bacteria? Okay, is it 23S ribosomal RNA, 16S ribosomal RNA, 5S ribosomal RNA, or ribosomal proteins? Okay, so ribosome, they are made up of ribosomal RNA. It has two subunits. Okay, so they are saying which component of the ribosome, okay, is involved in peptide bond formation in bacteria, which is a prokaryote. What is the answer? The option number A will be the correct answer. 23S ribosomal RNA. This is the correct answer. This acts as a catalyst for the formation of peptide bonds. Next one, a frameshift mutation occurs when? Option number A, a single nucleotide is substituted, altering one amino acid. Option number C, three consecutive nucleotides are inserted or deleted. Option number C, one or two nucleotides are inserted or deleted, disrupting the reading frame. Option number D, a stop codon is placed by an amino acid coding codon. So what will be the correct answer? Okay, here the answer is option number C. Okay, in frameshift mutation, what happens? In frameshift mutation, there is a change in the reading frame because of addition of one or two nucleotide or deletion of one or two nucleotides. Okay, so the reading frame will change, and now the new reading frame, it will code from some another amino acids. Okay, so the entire polypeptide will change. Now next question, which of the following is a stop codon in genetic code? Options are AUG, UUU, UAG, or UAC. What is the correct answer? The option number C will be the correct answer here. UAG is the correct answer. Next one, genetic code is considered degenerate because one codon codes for multiple amino acid. Multiple codons can code for the same amino acid. Then codons are read without punctuation. And option number D, the code is nearly universal across the organism. What is the correct answer? Option number B with the correct answer here. Multiple codons can code for the same amino acid. This is known as degeneracy of the genetic code. Okay, so we have 64 codons, but we have only 20 amino acids because multiple codons, they can code for same amino acids. Next one, which application is not associated with DNA fingerprinting? Okay, they're talking about the application of DNA fingerprinting. First is forensic identification, second is paternity testing, third is determining blood group types, and option number D, studying the genetic diversity in the population. What is the correct answer? Option number C is the correct answer here. Determining blood group type, this is not the application of DNA fingerprinting. Okay, DNA fingerprinting can be used in to solve the crime cases. Okay, for paternity testing. Okay, studying genetic diversity in the population also. Okay, but not for determining blood group types, whether the blood group is B positive or A positive. This is not the application of DNA fingerprinting. Next one, what is the significance of using PCR in modern DNA fingerprinting? Okay, option number one, it replaces restriction enzyme. Second is it amplifies DNA, enabling analysis from a single cell as well. Option number C, it eliminates the need for probes, and option number D, it directly visualizes VNTR without the electrophoresis. What is the correct answer? The option B is the correct answer here. See, PCR is polymerase chain reaction. Okay, so with PCR we can amplify the DNA. Clear? So we can make billions of copies of from a single molecule of DNA. Clear? So PCR is used for amplifying the DNA. So DNA can be amplified even from a single cell. Okay. The next one, which step in DNA fingerprinting involves transferring DNA fragments to a synthetic membrane? Okay, so options are electrophoresis, restriction digestion, southern blotting, or autoradiography. What is the correct answer? Okay, the option number C will be the correct answer, southern blotting. Okay, so in southern blotting, what is happening here? The DNA fragments, they are transferred to the synthetic membrane like nitrocellulose membrane. Okay, clear? Next one, in DNA fingerprinting, why do VNTRs produce distinct band patterns for the individuals? Okay, so VNTRs, these are the basis of the DNA fingerprinting. Okay, so let's have a look at the options. So options are they code for unique proteins. Option number B, they copy, their copy number varies between chromosomes and the individuals. Option number C, they are inherited only from the mother. Option number D, they are located in mitochondrial DNA. What is the correct answer? Option number B is the correct answer here. Their copy number varies between chromosome and individuals. So VNTR is variable number of tandem repeats. So certain sequences, they are repeated again and again, and these sequences are adjacent to each other. Tandem means they are adjacent to each other. Okay, but their copy number varies between the chromosomes also and among the individuals also. So this is the basis of DNA fingerprinting. Okay, clear? So option number B will be the correct answer here. So with this, we have discussed the mock test also. Clear? I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.